Hi friends, you're back with me, Professor Girish Kukreja. As I told in my earlier video that today we'll be talking about one interesting group of organisms. Like we are in a series where we are discussing some unexplored, I'll say rather recently explored group of organisms or some different what you call as aspects of microbes. It may be your algae, bacteria, fungi. Many of the times like in our academic studies we focus more on bacteria. Today I'll be talking about a group of organisms which probably has had a very complex or confusing taxonomic history. This group of organism, as I earlier also told, that it belongs to the group of fungi. These are sometimes popularly called as the most curious fungi. Because one of the genera which was discovered first, that is your Nosema, it was first put along with your fungi and your yeast, your mold. But later on, when it was studied more, they were put in different class of your protozoa. They were put in your Nidophora, then they were put in your Archaeozoa. And then later on, when molecular methods of taxonomy they creeped in. So when we started knowing the organisms in detail based on their RNA, what you call as your sequences, based on the sequences which were obtained of alpha and beta tubulin, it was known that this class of organisms, that is your Microsporidia, they are more closely related to these fungi. Yes, so they are closely related to your fungi or at least a sister group of these particular organisms which we call as your fungi. So these particular organisms, they are unique in various ways. You can say that the spores which they produce, which you can see here, are some of the smallest known eukaryotic organisms ranging in size of around 1 to 40 micrometer. So you will find that these organisms, as I told, being eukaryotic, when we imagine about eukaryotes, we imagine an organism having all those cellular structures or you can say subcellular organelles, but here you will find that they have very few organelles. The major powerhouse of the cell which we talk about, that is your mitochondria, which defines all what you call as heterotrophs, even autotrophs. Autotrophs are not defined by mitochondria, but yes, we know that they have these powerhouses, that is mitochondria. These group of organisms, they lack them. They do not have mitochondria, they have mitosomes, right? So they do not have mitochondria, they do not have peroxisomes, they do not have Golgi complexes. Sometimes then it becomes like how do they survive? They are obligate intracellular parasites. The term which we probably restricted up till now only to your viruses. Up till now we used to say that these viruses are obligate intracellular parasites. But this group of organisms, they are also obligate intracellular parasites. Almost all group of animals, they are infected by these. Major you will find. Recently, a large group of organisms are found to infect coleopterans on a very large scale. Probably most of the insects, they are being infected by these particular Microsporidia. Probably that makes them a potential candidate to be used as a biocontrol agent. So if you want to kill a particular pest or an insect, you can count on these Microsporidia maybe in coming near future. So these are obligate intracellular parasites. Yes, not only in your insects, but they are also parasites in humans. So we have to be like very careful. So they are parasites to humans, they are parasites to fishes, they are parasites to crustaceans. So all group of organisms, I told, your insects, your fishes, your humans, all of them, they are what you call as under the threat of this particular Microsporidia. It leads to a rare disease which we call as your microsporidiosis, right? Very rarely it will develop into that. Most of the infections are not lethal, but then yes, your productivity, vigor and many other things, they can be what you call as affected. In fishes and all, especially you will find in certain groups there is formation of xenoma and many other complications. In humans, if we are talking about, I told there are many different species which infect humans also, but in humans also they will infect only those which are immunocompromised. Especially we know that when in a particular person is infected with HIV AIDS, the count of CD4 plus cell or T helper cell, what we call, if that goes below 150, then probably there is a great threat of the infection of this Microsporidia. Some of the common species which are found as human pathogens include your Enterocystozoan bionoaceae, causing pneumonia, causing diarrhea, depending on from where this organism has entered. So if it has entered through inhalation, it will cause pneumonia. If it has entered through what you call as your digestive tract, if you have consumed these particular spores, they will reach your intestine and they will cause diarrhea. Some other species include your encephalatozoan cuniculi. So these are responsible for causing encephalitis, your nephritis, inflammations in your kidney. So these organisms are represented by these spores. Like as I told, some of the smallest known eukaryotic organisms, you can say, having a range of 1 to 40 micrometer. So especially the organisms which actually infect the mammals, they are hardly 1 to 4 micrometer. So they are very small. Matlab, imagining such a small eukaryotic organism itself is like amazing. So these particular spores, they have a typical structure. They have a typical exospore. They have a median which is sometimes known to contain, most of the times known to contain chitin, defining them as a fungi, and then the endospore. What makes them different here is that they have their special infective organs which we call as the polar filament, also referred to as the polar tube. So this particular polar tube, actually, this you can say is the major what you call as organ through which it is going to actually infect the host cells. So this particular organism, you can see it has this exospore, it has a median, it has an endospore, it has this polar filament, and it has this anchoring disc. So whenever it encounters a particular host, so inside the host, inside the gut of the insect or any other host, whatever it is specific for, so there will be what you call as imbibition or what you call as accumulation of water and other fluids here. So because of that osmotic pressure in this posterior vacuole, this particular polar filament which is there, it will be extruded out. So this membrane, it would be broken. So the what you call as this membrane is actually tough, but then the thinner part of it, it is what you call as broken, and this particular filament, it will extrude out and it will enter into the protoplasm of the cell, so of the host cell obviously. And once it enters into the host cell, all the material enters this tube and then it is ready to infect the host. And as I told that they are obligate intracellular parasites, they will capture the entire machinery of the host cell, and now the host cell is you can say under the total control of these particular organisms. They show different variations in their life cycle. Matlab, certain groups you will find that they are showing only asexual phases, whereas there are certain groups which will show sexual phases. A typical plasmodium kind of a thing you can see, where there is multiple fission and multiple spores, they are formed once inside the host. So there is a huge variety, I can say. In these particular spores also, matlab here for simplicity we have shown a typical oval or egg-shaped spore, but then you will find that having a rod-shaped spore is also not uncommon in these particular groups. So especially sometimes the size and the shape of these spores is used to define or classify these particular organisms which we call as Microsporidia. Many of them you will find that they are hyperparasites. Matlab, we are known to Digenians, the parasitic flatworms which infect many mollusks and what you call as vertebrates also. So these particular parasitic flatworms, they are actually parasites. They are dependent on these vertebrates and your mollusks, but these Microsporidians, they are parasites on those parasites. So Digenians, they harbor these Microsporidia. So those particular organisms we refer to as what is called as your hyperparasites. The genome size, as I told, that they are sacrificing many genes, like they don't have mitochondria, they don't have peroxisomes, they don't have Golgi complexes. Their RNA genes also are found to be very small. So you can say one of the smallest known genomes, nuclear you can say, or eukaryotic genomes, is that of these particular Microsporidia, with the size of around 2.5 to 11.6 MB, coding for around 1848 to 3266 proteins, which is very near to some of these particular bacteria. So these are a group of organisms which are you can say a focus now. Matlab, we want to study them because they are more what you call as harmful to those patients which are immunocompromised. They can lead to serious disorders. There was a particular say classification status, like earlier it was thought that they are very primitive. Matlab, they came very early during the evolution. But now people are coming up. There is a school of thought that which says that they have now actually evolved, and they have lost these particular genes of mitochondria, or they have lost the mitochondria, they have lost the Golgi complex, and they are now evolving. So some group of organisms or some group of individuals, they assume that this is a more you can say evolving or a more modernly evolved organisms which is now actually what you call as losing these particular genes and trying to cope up or trying to adjust to this new environment. So this particular group is a recent group of study where people are trying, placing them from this protozoa to fungi. It happened recently at around mid 2000s. Many texts will sometimes also cite them still as a protozoa, but with modern molecular methods we now know that they belong to the group of what is called as your fungi. So this intricate group of organisms can be like harmful at the same time useful. As I told that they are now known to infect many insects, so we can count them as a major biocontrol agent. So stay tuned with me, Professor Girish Kukreja, for more in microbiology, and let us know and explore more about this wonderful world of microbes. I request you all that if you also want to know about a particular group of organisms, or if you want to share a group of organisms, you want to share some novel activities of the organisms or some novel bacteria, protozoa, fungi, algae, cyanobacteria and many more, you can please contact us and we can explore this world more. Thank you so much and stay tuned.